Hi students, today let me talk about urea cycle. Very important essay question, the first and foremost essay question from amino acid metabolism, right? Essay question, short notes, clinical scenario, MCQ, everything is from urea cycle, right? So first we are going to introduction about the urea cycle or urea. Before going to the description about the urea and ammonia, first you have to mention about the normal value of blood urea, that is 20 to 40 milligram per deciliter, right? Don't forget. Second one is all the nitrogen from amino acid converted into ammonia. We know ammonia is very toxic, that should be converted into non-toxic urea, which will get excreted. This is the concept of urea cycle. Why urea cycle occurs in our body? All the nitrogen compounds from amino acids produces ammonia. This ammonia is very toxic to our body. So this should be converted into non-toxic compounds like urea and get excreted. So the urea is end product of protein metabolism. Very simple. It is synthesized from liver, that is urea cycle occurs in liver and excretion done by kidneys. Next one is urea, you know the formula NH2C double bond O NH2, right? That is two amino groups, one carbon skeleton. These two amino groups are derived from two separate compounds, like one amino acid is aspartate, another one is ammonia NH3. This is very important MCQ. Urea formula which is having two amino groups where these two are derived from different compounds. One is from aspartate, another one is from ammonia, right? Carbon skeleton is synthesized from CO2 molecule, which is usually derived from TCA cycle and biological ETC, electron transport chain, and biological oxidation. Next one is we have actually in the urea cycle 5 reactions. First two reactions are taken place in mitochondria, whereas the remaining 3 coming in cytosol. This is very important MCQ. First two, it starts with the mitochondria and ends with cytosol. Okay. Next one is the regulatory enzyme for this urea cycle is CPS1, that is carbamoyl phosphate synthase 1, right? We have CPS2 also. So you should know the difference between CPS1 and 2. CPS1 is the mitochondrial enzyme which is used for the urea synthesis, whereas CPS2 is a cytosolic enzyme which is used for the synthesis of pyrimidine, that is nucleotide. This is again important MCQ point. And the last one is this cycle found out by Krebs and Henseleit. Krebs is the boss and Henseleit is the student medicos. They found out the cycle. You know the Krebs cycle, that is TCA cycle, already found by this scientist. So Krebs Henseleit cycle is also known as urea cycle. Okay. Now we are entering into the urea cycle reactions or metabolic pathway of urea cycle. Okay. For the first reaction, we require two important compounds, carbon dioxide molecule and ammonia, which is derived from cytosol. Actually, first two reaction takes place in mitochondria. For this mitochondrial initial reactions, we require one molecule of carbon dioxide and one molecule of ammonia, which is synthesized derived from cytosol. Okay. So, with the help of CPS1, this carbon dioxide molecule and ammonia synthesizes carbamoyl phosphate. This is the first step. Okay. For this CPS1, we require some energy, that is 2 molecules of ATP, which will be converted into 2 ADP and inorganic phosphate. The important role for the first step is NAG, N-acetyl glutamate, which is the positive modifier or activator of the CPS1. They only increase the synthesis of carbamoyl phosphate, that leads to urea cycle synthesis increase, right? That is why CPS1 is a regulatory enzyme. For this CPS1, we require N-acetyl glutamate, which is the positive modifier or activator, right? This carbamoyl phosphate, second step, combined with the ornithine, produces citrulline and inorganic phosphate. Here the enzyme is very simple, carbamoyl and ornithine. So ornithine transcarbamylase is the second step enzyme which releases citrulline. So the first product is carbamoyl phosphate, second product is citrulline. Okay. Here what happens is this ornithine is derived from cytosol to mitochondria, that is enters into mitochondria. Whereas this citrulline from mitochondria to cytosol. This step is very, very important. After entering into the cytosol, this citrulline is synthesized arginosuccinate with the help of arginosuccinate synthase. Here also we are requiring two molecules of ATP, that is one for AMP and one for inorganic pyrophosphate. Here the important compound which is helped for the synthesis of arginosuccinate is aspartate, right? So citrulline combined with aspartate with the help of arginosuccinate synthase and two molecules of ATP produces arginosuccinate. This is the third step. Fourth one is this arginosuccinate releases two important compounds. One is fumarate, another one is arginine. Here the enzyme is arginosuccinate, so arginosuccinase. Another name of this enzyme is arginosuccinate lyase. Okay, important MCQ. Here what happens is, if you are getting fumarate means, this will be the intermediate of TCA cycle product. So automatically they enters into TCA cycle. Okay. Another one is they release arginine. What is the importance of this arginine is, this only releases the urea along with the ornithine. This step is very, very important. Most of the students forget to mention about the urea. Actually your question is urea cycle, but you people forget about to mention the urea name. Arginine releases first urea and then ornithine with the help of arginase. This is very, very important step for your urea cycle. Again this ornithine ready for the another reaction of urea cycle. So totally we are having 5 reactions. First 2 occurs in mitochondria, remaining 3 occurs in cytosol. The last step, that is arginine releases urea as well as ornithine, is very, very important step. Okay. So this is how you have to draw the urea cycle for your essay question. Okay. Right. Now we are coming to the part 2, that is second part of urea cycle. Okay. Already we discussed about the introduction and reactions or metabolic pathway of urea cycle. Now, how it is regulated? What are all the points for the regulation? Very simple, only 2 points. Number 1 is substrate availability. For any pathway in biochemistry, first the availability of the substrate is very, very important. Carbohydrate, protein, lipid, nucleic acid, anything. If the substrate is available means the pathway will be continued. If there is no pathway or shortage of pathway, the pathway won't occur. So like that, if you take more protein diet, there will be more urea cycle in urea synthesis. If protein is less means urea cycle also will be less. Second one is CPS1 is the rate limiting enzyme for urea cycle in the step 1. For this CPS1, we have one allosteric activator, that is NAG, N-acetyl glutamate. This is actually positive modifier which is synthesized from 2 compounds, glutamate and acetyl CoA. Sometimes they will be asking in some MCQs. NAG is synthesized from dash and dash, glutamate and acetyl CoA. What is the use of this NAG? This is the positive modifier of CPS1. This is the allosteric activator of CPS1, which will increase the synthesis of urea cycle. Okay. Next, what is the significance of this urea cycle? What is the use? First one is, from the urea cycle you are getting arginine, amino acid. This arginine synthesizes nitric oxide, very important short notes for you, and it is actually potent vasodilator and also muscle relaxant. Nitric oxide, vasodilator and muscle relaxant. Next one is, this cycle disposes two important compounds or waste products. One is carbon dioxide molecule, second one is ammonia. Carbon dioxide molecule enters into respiratory regulation of acid-base balance also. That is why third reaction is regulation of blood pH. So depending upon the availability or concentration of CO2 only, carbonic acid is synthesized in blood, H2CO3. Depending upon the concentration of CO2 and H2CO3 only, bicarbonate is formed, HCO3. So it will be useful for the regulation of blood pH, urea cycle. Right? This is the significance for urea cycle. Next one is, what is the energetics for urea cycle? For any biochemistry pathway, if there is any energetics, means you should underline and memorize the ATP molecules. That is, number one, how many ATPs are utilized. Number two, how many ATPs you are giving for the cycle. Number three, no ATP synthesis. These are all the 3 points you have to mention about the energetics. Here what happens is, 2 ATPs for CP synthesis, that is carbamoyl phosphate synthesis we are giving, we are donating. One more 2 ATPs, another 2 ATPs for arginosuccinate synthesis. So we are giving 4 ATPs molecules for the synthesis of urea cycle. Right. Actually, we are having one intermediate product of TCA cycle, that is fumarate in this urea cycle. This fumarate enters into TCA cycle, that is conversion of malate, oxaloacetate, like that. In that conversion, they will produce one molecule of NADH. We already know FAD and NADH produces how many ATPs? NADH 2.5, FAD 1.5 releases in the electron transport chain. So this fumarate releases in TCA cycle 2.5 ATPs. Already we are giving 4 ATPs. So 4 minus 2.5, 1.5 ATPs. Right? So energetics of urea cycle means, answer is very simple, 1.5 ATPs produced. Whereas how many ATPs are required means 4 ATPs. You got it? First one to second one to. Very carefully you have to read about the energetics. How this 4 minus 2.5 is equal to 1.5, you should mention very clearly one by one. Okay. Next, why this urea cycle is known as Krebs bicycle? So actually this fumarate linked with TCA cycle. How? In TCA cycle, fumarate enters into TCA cycle and converts into malate, finally converted into oxaloacetate. This is the TCA cycle step. In this oxaloacetate, they will go for a transamination reaction and releases aspartate. And this aspartate goes for the urea cycle. So this is the step for the interconnection between TCA cycle and urea cycle. That is why this is known as Krebs bicycle. Sometimes recent out questions like that in MJ University or Bharat University, they will be liking this. Why urea cycle is called as urea bicycle or Krebs bicycle? That time, the only one word which will come to your mind is fumarate. Fumarate enters into TCA cycle. TCA cycle, oxaloacetate produces and goes for transamination, and they will be releasing aspartate. Aspartate again enters into urea cycle. Like that, you have to link the two pathways, that is urea cycle and Krebs cycle. Okay.